Last group chapter 1, sexual reproduction of flowering plants. Then if I will talk about the sexual reproduction. Sexual reproduction in plants was discovered by Rudolf Jacob Commercius in 1694. Then firstly I will talk about the sexual reproduction. Sexual reproduction in plants was discovered by Rudolf Jacob Commercius in 1694. Sexual reproduction refers to the fusion of male and female gametes, forming zygote that develops to form the new organism through embryogenesis. The gametes are formed by the same individual or by different individuals of the opposite sex. Now, what is zygote? Zygote is the vital link that ensures the continuity of species between organism of one generation to the next. Embryogenesis is the process of development of embryo from the zygote. Then now I will talk about the structure of the flower. Structure of the flower. Then the floral axis bearing the floral organs is called the receptacle. Now here the structure of the flower, and this region is known as the receptacles. Then receptacles, the floral axis bearing the floral organs is called a receptacle, and therefore the various floral word taken in distinct word. The outermost sepals and part are the petals. It is androsium made up of andros stamens. Now in the flower, morphology of the flower consisting the sepals and inner to this a whorl of corolla made up of petals. Colorful part, it is petals. Next to corolla toward the inside is androsium. In the petals present the androsium. These are the androsium made up of stamens representing male sex organ. Stamens are the male sex organs of a flower. The innermost world, सबसे इनरमोस्ट की तरफ अगर देखने को मिलता है तो वो गाइनोसियम. Then gynosium consisting of carpels representing female sex organs. Calyx and corolla are called accessory world. Calyx and corolla, it is the accessory world. Whereas androsium and gynosium androsium and gynosium, it is the essential world. It is the essential world due to their direct involvement of sexual reproduction. Now When sepals, when sepals and petals are not differentiated, the undifferentiated world is called a perianth, and each member of perianth is called a tepal. Undifferentiated world of flower, it is known as perianth, and each member of perianth is called tepal, is called the tepals. Okay, each stamen, well, the stamen, each stamen has two parts, a filament and mostly bilobe anther. Each stamen contains two parts. Number one is a filament, and number two is a bilobe. Each lobe dithecous. Each lobe contains dithecous. Each lobe contains two microsporangia or pollen sacs. Each lobe, it is bilobe. Each lobe contains microsporangia or pollen sacs, having a total of four microsporangia in anther. Now here, if I will take a transverse section of the anther, then it is made the microsporangium. There are four microsporangium. Now, which is family it's belong? Its family is a Malvaceae. Its family is a Malvaceae. Malvaceae family. Okay. In members of family Malvaceae and in some other plants, monothecous bisporangiate anthers are present. Each carpel consists of three parts. Each carpel consists of three parts, namely, then firstly I will talk about the carpel. Carpel, it is a female sex organ of the flower. Female sex organ of flowers. Then carpels are divided into three parts: ovary, style, and stigma. Ovary, style, and stigma. Okay. The ovule or megasporangium develops within ovary attached to the placenta with the help of cuticle. I will talk about in next. Then I will talk about the development of the microsporangium. It means pollen sac. Development of microsporangium, pollen sacs. Development of pollen sac. It means microsporangia, microsporangia. तो सबसे पहले मॉर्फोलॉजी के बारे में बात करते हैं तो इट इज़ अ microsporangium and this is a transverse section of bilobed anther. Anther is a male sex organ of the flowers. फिर सबसे पहले जो फॉर्मेशन होता है एपिडर्मिस, इसके बाद इंडोथीसियम, देन मिडिल लेयर, then tapetum and then microspore. And sporogenous tissue, it functions. What is function? I will discuss now. The cells of sporogenous tissue. The cells of sporogenous tissue, after separation from each other, function as pollen mother cell. It means PMC, pollen mother cell. It is already deployed in nature to n number. And can also be called meiocytes. Each PMC undergoes meiosis to form four haploid pollen grains or microspores arranged in a form of tetrad. During meiosis in PMC, two patterns of cell wall formation can be seen. The cell wall formation is of two types. Successive. Number one is successive. After the first meiotic subdivision, a callose wall is formed separating the two nuclei, leading to the formation of the dyad. The two cells of a dyad undergo second meiotic subdivision to form a tetrad of four cells. The callose wall is again formed after second meiotic division. The type of tetrad is called isobilateral and is of common occurrence in monocotyledons and polypetally of dicotyledons. Okay. And next one is a simultaneous. In simultaneous type, the first meiotic in simultaneous type, the first meiotic subdivision is not followed by wall formation, eliminating the formation of a dyad. After second meiotic subdivision, callose walls are laid in such a manner to give rise to tetrahedral tetrads. Such tetrads are common to gamopetally of dicotyledons. It is the function of the sporogenous tissue. Now next version is anther wall. The mature anther wall shows an outermost single layer epidermis followed by a layer of endothecium. Endothecium. The matured anther wall shows an outermost single layer epidermis followed by a layer of endothecium, 2 to 3 layers middle layer. Okay, and a single layer innermost. It is a tapetum. It is a tapetum. Then, firstly, and the matured anther wall shows an outermost single layer epidermis, and then outermost single layer in epidermis followed by a layer of endothecium. Tapetum having as it supplies material for the the development of pollen grains pollen grains or it may serve as transport channel through which material can reach the then the amyloid type of tapetum is responsible for producing callase enzyme for the release of pollen grains of a tetrad by dissolution of callose wall. At the time of pollen wall formation, tapetum plays a vital role through obese bodies coated by sporopollenin, helping in external thickening of exile. Sporopollenin is resistant to physical and biological decomposition. Are preserved for long periods during fossilization. Another function of tapetum is transfer of pollen kit material and trifeed the pollen surface. Now next, tapetum transfer to transfer of pollen kit material and trifeed to the pollen surface. Pollen surface. Then tapetum function is the transfer of pollen kit material and trifine to the pollen surface. Then I will talk about the pollen kit. Pollen kit is the most oily layer, oily layer forming forming a thick coating a thick coating of pollen surface of pollen surface of many insect pollinated species. Pollinated species. The thickness, order, and color of pollen grains are due to pollen kit material. Now next point is the thickness, order, and color of pollen grains are pollen grains are due to pollen kit material. Due to pollen kit material. This material is mainly composed of carotenoid and flavonoid pigments, imparting yellow or orange color to the pollen. The following functions may be attributed to pollen kit actioning as the insect attactant, protection of pollen grains against the damaging effect of UV radiation. And next one is the tryphene. I will discuss now here. Pollen kit now. Tryphene. Tryphene, it is composed of proteins and it is composed of proteins and hydrophilic substance and pollen energy. It is function of tryphene. Now here I will discuss. This is a microspore mother cell. Microspore mother cell. Then sporogenous tissue after separation. After separation from each other function as pollen mother cell and can also be then it is a gametophyte. It is a divided here. Now here डिहाइड्रेशन में सारे सेल्स जो होते हैं यहाँ पे अलग अलग हो जाते हैं तो and during meiosis in PMC, two patterns of cell wall formation can be seen. The cell wall formation of two types. Formation of cell wall are two types. First one is successive and second is the simultaneous types. Okay. Next, I will talk about the allergic pollen grains. After dehydration, pollen grains are dehydrated here and pollen grains structure. You can see this.